All right, chapter 19, renal physiology. And this is going to be the beginning of the last bit of information for your final. Don't forget your final is not comprehensive. It's just going to cover these last few chapters. We'll start with chapter 19. We'll start talking about the kidneys and renal physiology. All right, these are the 6 functions of the kidneys, and they're pretty easy to understand. This is a pretty easy read. First, regulation of extracellular fluid volume and blood pressure. Well, we talked a little bit about blood pressure in the cardiovascular system. Whenever the blood vessels would vasoconstrict, then it would increase blood pressure, and then whenever the arterioles would vasodilate, then it would lower the blood pressure. Well, the kidneys can also help regulate the blood pressure by regulating how much fluid leaves the circulation and goes into the bladder. So that kidneys can conserve more fluid in order to raise your blood pressure when it gets too low, or it can put more fluid into the bladder in order to lower the amount of fluid in your cardiovascular system, which will then lower your blood pressure. All right, then the kidneys are used for regulation of osmolarity. It can control which ions are going to leave the circulation and which ones it's going to reclaim and put back into your circulation. Kidneys are also used for your ion balance. That kind of goes back up here with osmolarity anyway. And then the kidneys are used to regulate your pH. How does it do that? Well, it can either reabsorb hydrogen ion from the filtrate that's leaving the circulation, or it can take hydrogen ion and secrete it back into the filtrate to get rid of it. So if you retain more hydrogen ion, it will make you more acidic. So the reason why your kidney will retain more hydrogen ion is because you became alkaline. Your pH started to rise, so it reabsorbs and reclaims some of the hydrogen ion back into the filtrate or back into the cardiovascular system in order to lower your pH. And then the opposite of that, if we become more acidic, that means it can take the hydrogen ion, put it into the filtrate so you can excrete it with the urine. So as you're losing hydrogen ion, it'll help raise your pH to make you more alkaline if you're too acidic. And then the function that most of us identify with the urinary system would be excretion of wastes like urea and uric acid. And then there are some hormones that come from the kidneys, like erythropoietin, and also renin comes from the kidneys. And we'll look at the renin-angiotensin system later in this chapter. All right, I just skipped over a few slides because it's a review of the anatomy of the kidney. So you can read through those slides, and I'm just going to talk about some of the pictures that we see here. We can see you have 2 kidneys, one on the right, one on the left. One of them is slightly lower than the other. The right kidney is slightly lower because the liver is right above it, so it pushes it down just a little bit compared to the right kidney. Blood goes into the kidney through the renal artery, and then it circulates around in the kidney. We're about to look inside the kidney. There's the functional unit of the kidney is called the nephron, and we'll look over the parts of the nephron here in a minute. So your entire blood circulation will go into the kidneys through the renal artery, and then they will leave through the renal vein. And then the filtrate that is made by the kidneys and the nephrons will then travel down through a tube called the ureter. And the ureter travels all the way down through the abdomen into the pelvis and ends up at the bladder, and the bladders will temporarily store the urine until you can get rid of it. There's another tube that goes from the bladder to the outside, and that would be the urethra. When we look inside of the kidney, this is a sagittal section through the kidney, we separate it into a cortex and the medulla. The cortex of the kidney is in the outer part of the kidney, and then where we see those pyramids, the renal pyramids, that would be the medulla of the kidney. The functional unit of the kidney is the nephron. If the nephron is just in the cortex, then they're called cortical nephrons, but some of them actually dip down into the medulla as well, and those are called juxtamedullary nephrons. Most of the nephrons are cortical nephrons found in the cortex. If we review the blood flow into and out of the kidney, blood goes in through the renal artery, and then as soon as it goes into the renal pelvis, it becomes the segmental artery. And then the segmental artery, when it goes into between the lobes of the kidney, it's called the interlobar artery. So I'm now going to go to this picture. Here's the interlobar artery. And when the interlobar artery curves around the base of the pyramid, it becomes the arcuate artery. And then the arcuate artery goes up into the cortex. And this is called the interlobular artery or cortical radiate artery. And then after that, it goes into the afferent arteriole. And the afferent arteriole is where we generally start whenever we talk about blood going into the nephron. So blood goes into the nephron through the afferent arterioles. Then it goes through the glomerulus, comes out to the efferent arterioles. And then the efferent arterioles become peritubular capillaries and vasorecta capillaries. Remember that the kidneys are a portal system because there are two sets of capillaries. First, there are the capillaries into the glomerulus, and then we have the capillaries of the peritubular capillaries in the vasorecta. And then whenever blood is leaving the nephron and leaving the nephron area, it comes back through the cortical radiate veins, and then it goes into the arcuate vein. The arcuate vein goes into the interlobular vein, and then the interlobular vein goes straight into the renal vein. There is no segmental vein, and then the renal vein leads to the inferior vena cava. On this picture here, we can see the cortical nephrons that are mainly in the cortex, and then the juxtamedullary nephrons that dip down into the medulla. And now we can see the peritubular capillaries. The peritubular capillaries stay in the cortex, and they surround the proximal and the distal convoluted tubule, and then the vasorecta. Those capillaries go around the nephron loop. There are two parts to a nephron. There's the renal corpuscle and then the renal tubule. I'm going to draw a line right here. Everything to the left of that line is part of the renal corpuscle. Everything to the right of that line is part of the renal tubule. So the two main parts of the renal corpuscle is Bowman's capsule. Bowman's capsule, if you remember, is in two layers. It's a parietal layer and a visceral layer. The parietal layer is on the inside, the visceral layer being the podocytes that surround the glomerulus. The podocytes are part of the filtration membrane that filtrate has to pass through in order to get into the space, the Bowman's capsule space. So the other part of the renal corpuscle is the glomerulus, and the glomerulus is the ball of capillaries. And if you remember, the afferent arteriole goes into the renal corpuscle, becomes the glomerulus, and then the efferent arteriole comes out of the glomerulus. All right, and then we have parts of the renal tubule. We'll start at the proximal part, which is going to be the proximal tubule. It's called the proximal convoluted tubule because it is closest to Bowman's capsule and it's not a straight line. So proximal convoluted tubule. If you follow that around with the light, so here we have the filtrate coming out from the glomerulus, goes into Bowman's capsule, and then it goes into the proximal convoluted tubule. And then the proximal convoluted tubule will then become part of the descending limb of the nephron loop. There's a thick limb and a thin limb. We're not going to worry about the difference between thick and thin, but this is the descending limb, and then it goes through the loop of Henle. Then it goes up through the ascending limb, and the ascending limb has both a thin and a thick portion as well. Then the ascending limb becomes the distal convoluted tubule. Distal because it's farther away from Bowman's capsule, and it is convoluted because it's not a straight line. And then eventually it joins with the collecting duct. So let me go back to my little marker. So the proximal convoluted tubule leads to the descending limb of the nephron loop. The descending limb of the nephron loop then circles around through the loop of Henle. And then as it goes up through the ascending limb, the filtrate enters into the distal convoluted tubule, and then the distal convoluted tubule leads to the collecting duct. I just want to point out that multiple distal convoluted tubules lead to the same collecting duct. And then the collecting duct goes through the renal pyramid and ends at the renal papilla, where it empties the filtrate into the minor calyx. If you have trouble remembering all these parts of the renal tubule and the renal corpuscle, I would probably try to keep that anatomy next to me as I go through these parts of the physiology, because if we can picture what's going on, that'll help us to remember the physiology. There are three processes that lead to urine formation. So these are the three main functions of the kidneys. First, there's going to be filtration. It filters stuff out of your blood. And then we have reabsorption. Reabsorption means taking some of the stuff that's been filtered out and putting it back into the circulation. And then we have secretion. Secretion is taking something from the circulation and putting it directly into the filtrate. So when you start thinking about these processes of the kidney, think about the direction that the fluid and the ions are going. For example, with filtration, fluid and ions are leaving the cardiovascular system, leaving the glomerulus, and going into the renal tubule because it's part of the filtrate. The filtrate will eventually become urine if it stays inside the tubule long enough. Then reabsorption. The direction of the fluid and the ions from reabsorption is going from the filtrate and going back into the cardiovascular system. And then secretion. The direction of the fluids and the ions in secretion is going from your circulation of the cardiovascular system and back into the filtrate. And now as a review, remember the difference between internal environment and external environment. Internal environment means it's inside the body. External environment is not considered to be part of the inside of the body. It's easy to think of that being outside, inside the skin. The stuff that's touching the outside of the skin is external environment. And the stuff that's on the inside of the skin would be the internal environment. What we forget about are the lumens of tubules, like the lumen of the alimentary canal. It's on the inside, the food stuff that you put in your mouth and you chew up and swallow. That's not considered inside the body and internal environment until it's been absorbed through a membrane. So the same thing with the lumen of the renal tubule. Anything that leaves the glomerulus, the filtrate that leaves the glomerulus, those ions, and the water that leaves is part of the external environment. So filtration, again, is moving fluid and ions from internal environment to external environment. And then reabsorption. Reabsorption is taking that fluid and those ions back into the internal environment from external. And then secretion is moving ions from the internal environment directly into the external environment of the renal tubule. Let's look at some numbers here. The filtrate that comes out of the glomerulus and it's caught by the renal corpuscle adds up to about 180 liters per day. And if you think about it, that is 92 liters. We don't have that much blood in our body. We only have about 5 liters of blood in our body. So where does the 180 liters come from? That's because most of it's going to be reabsorbed back into your body. We have to lose that much fluid whenever it's making the filtrate for the urine, but we're going to reclaim most of it back. The osmolarity of the filtrate is almost the same as plasma. Typically, plasma osmolarity is around 290, 280 milliosmoles. So the fact that this is 300 is not that big of a difference, but it is a difference, and that's because of the amount of fluid compared to the amount of fluid in the rest of the body. The proximal convoluted tubule is going to reclaim about 70% of that back into the cardiovascular system. That is part solutes and it's part water. Again, this is still about 300 milliosmoles. And we're going to look at a picture here in a minute, and we're going to follow it and visually see how the osmolarity changes as the filtrate moves through the renal tubule. All right, then the filtrate moves through the loop of Henle. It claims another 18 liters. But now the osmolarity has changed. The osmolarity is a lot lower. So then the filtrate that enters into the distal convoluted tubule and the collecting duct, it takes back about 1.5 liters of the fluid that comes out per day. You'll see that there's a wide range of osmolarities between 100 and 1200 milliosmoles, and that depends on the antidiuretic hormone, the vasopressin hormone, because that's going to open up water channels. So the more water channels that are there, then the more fluid's going to be reclaimed from the filtrate, and it makes the osmolarity go up. Remember, the higher the osmolarity, the more concentrated the solution is. The lower the osmolarity, the more dilute the solution is. So if you take the amount that is excreted, the amount excreted is going to be the amount that is filtered out through the filtrate, subtract the amount that is reabsorbed, and then you add back in the amount that is secreted, and that will give you the total amount that's been excreted through the kidneys. Once the fluid makes it past the collecting duct, it is only going to go through the ureter into the bladder, and nothing else can be reclaimed, reabsorbed, or secreted into the filtrate. Well, I don't know whether to laugh or cry, but I just spent about 5 minutes explaining this slide to everybody and realized I didn't press the record button, so yay me. This is a picture showing you the 3 different functions of the nephron. We have filtration right here. Filtration only occurs at the renal corpuscle. That is where we have filtrate, leaving the glomerulus and then going into the renal tubule. That only occurs at the glomerulus. Let's look at some of the numbers and get the concepts. About 180 liters are going to leave the glomerulus during the day. So that is per day, not per minute or per hour, but for the entire day, 180 liters of fluid and ions are going to leave the glomerulus and go into the renal tubule. All right, the osmolarity is about 300 milliosmoles, which is about the same as the filtrate of the plasma. All right, and then we continue around. We go through the renal tubule, and then we go into the proximal convoluted tubule. Now we'll see that there are 2 other arrows here. We have the green arrow, which means reabsorption. So the direction of reabsorption is going from the lumen into the blood, going from external environment to the internal environment. And then we have the blue arrow at the proximal convoluted tubule, and that is secretion. And that is moving ions from the blood to the lumen. All right, we lose 180 liters per day, but when it comes to secretion only applies to ions, not fluids. We don't put fluids back into the lumen. We just take stuff out of the lumen. All right, so we go through the proximal convoluted tubule, reabsorbing, secreting, and then we go into the descending limb of the nephron loop. And you'll see there's only 1 arrow here. It's the green arrow, it's reabsorption. So reabsorption is moving, we're still moving stuff from the lumen into the vasa recta now, because we're talking about the nephron loop. The nephron loop has the vasa recta that surrounds that, and it can pick up anything that's being reabsorbed. All right, let's look at these numbers now. So when we go through the descending limb, it reclaims about 54 milliliters per day. And the osmolarity is about 300 milliosmoles, so it's still the same. So that means to us that this reabsorption process, at this point right here, where we start into the descending limb, we're losing ions and fluid at the same rate. It's keeping the osmolarity the same. All right, and then the fluid goes around the nephron loop, and now we're in the ascending limb. Ascending limb again only has 1 arrow, reabsorption only, so no secretion occurs from the nephron loop. All right, so when we get to the end of the nephron loop, let's look at these numbers. In the ascending limb, we have reclaimed about 18 more liters from the fluid, and now the osmolarity is 100. So osmolarity is lower. So what does that mean when you think of the concentration of the solution? The more concentrated the solution, that means the more solid particles per water particles. 300 milliosmoles at the descending limb. Now we're at 100 milliosmoles. That means osmolarity is lower. That means it is more diluted. So we're losing more ions right here in the ascending limb than we're losing fluid. So we've lost more ions and fluid. And then we go into the distal convoluted tubule. And at the distal convoluted tubule, we have 2 things going on. We still have reabsorption, and now we have secretion starting back up. Reabsorption is going from the internal environment to the external environment, and then from the internal environment to the external environment for secretion. All right, and then we're going to continue down. All right, we're going to go through the collecting duct. Once it gets past the collecting duct, it's going to go straight into the ureter, which will then go into the bladder. Let's look back up here at the collecting duct. We have reabsorption occurring. Reabsorption is going from external environment to the internal environment. And then we have secretion, moving ions from the internal environment to the external environment. And then we continue down. Now let's look at these numbers at the very end of the collecting duct, see how it differs. By the time we reach the collecting duct, the fluid that's going to leave and become part of the urine is going to be about 1.5 liters per day. And let's look at the range of the osmolarity. The range of the osmolarity is anywhere between 50 milliosmoles to 1200 milliosmoles. So it could be very diluted, which would be the lower osmolarity, or very concentrated, which would be the higher osmolarity. And that comes down to how much antidiuretic hormone or vasopressin is present. And we'll get to that later. This is a pretty good table to look at. What happens in different segments of the nephron. So, for example, at the renal corpuscle, that's filtration only, and it's going to be mostly protein-free plasma. What does that mean? That means the proteins stay in the circulation. If you have protein in your urine, then there's something wrong with the kidneys. And then at the proximal convoluted tubule, that's where you have isosmotic reabsorption. What does isosmotic mean? That means they have the same osmolarity. So the tissues that are surrounding the proximal tubule and the fluid inside of the proximal tubule, they have the same osmolarity. And then at the loop of Henle, we have reabsorption of ions and excess water. And we start to dilute the fluid in the lumen. So we're actually reabsorbing more ions than we are fluid in the loop of Henle, which is why the osmolarity goes down. Osmolarity inside of the loop of Henle, by the time we get through it, is around 100 milliosmoles compared to the 300 before. And then at the distal nephron, we have reabsorption of ions and water. And then this is where we're going to look at pH balance by either secreting or reabsorbing hydrogen ion. Now this is a simplified view of what we just looked at, the process of filtration that occurs right here between the glomerulus and the renal corpuscle, and then reabsorption, the direction going into the internal environment from external environment, secretion going from the external environment into the internal environment. And then we have this little formula down here. You take the amount filtered and you subtract the amount reabsorbed, and you add back in the amount secreted, and that will give the total amount of solute that has been excreted. All right, so the filtration fraction, that is percent of the renal flow that filters into the tubule. There are three things that you have to take into account when it comes to the filtration fraction, and it's going to be three parts of the filtration barrier. So this is the filtration membrane. The parts of the filtration membrane include the glomerular capillary endothelium. So that is the glomerulus, the capillary that's inside of the renal corpuscle. You take that endothelium. And then there's a basement membrane, and then there's going to be the podocytes. There are filtration slits of the podocytes. Podo means foot. Cyte is cell. So these are cells with little foot processes, and they create slits called filtration slits. So the fluid has to go through the glomerular capillary endothelium, through the basement membrane, and then through the filtration slits of the podocytes. The filtration fraction is around 20%. 80% of the circulation stays inside of the glomerulus, it stays inside of the cardiovascular system. So the 20% that is filtered out, more than 19% of it is going to be reabsorbed back into the system, which means that less than 1% of the filtrate that is from the filtration fraction is actually going to become part of the urine, and it's excreted to the external environment. This picture is showing you the renal corpuscle and also the parts of the filtration membrane. All these purple cells we see, those are the podocytes, and you can see the little feet to go to the side and they create these filtration slits. The afferent arteriole, blood coming in, and then once it gets into the glomerulus, we lose the filtrate. We lose 20% of it as the filtrate, and then the blood exits through the efferent arteriole. All right, so this picture shows you the filtration membrane again, but it's an artist's drawing. The filtrate goes from the capillary lumen through the glomerular endothelium, the basement membrane, in between the filtration slits to become part of the filtrate. There are three pressures to take into account when we talk about filtration. Those pressures are the capillary hydrostatic blood pressure. That is your blood pressure, or that's the pressure of the blood going through the glomerulus capillary. Now it's a hydrostatic pressure, and if you remember back to the chapter on capillaries, we talk about hydrostatic pressure as forcing fluid out into the tissues that are surrounding the capillaries. So, capillary hydrostatic pressure favors filtration. It's around 55 millimeters of mercury. The specific number does not matter, just the overall concept. But for now, we got to think of it this way. That 55 millimeters of mercury of pressure from hydrostatic pressure is forcing fluid out into the renal corpuscle. The other pressure to take into consideration is the colloid osmotic pressure inside of the capillary. And if we remember back to the chapter on capillaries, colloid osmotic pressure is created by the proteins of the plasma, more specifically the albumin. The albumin is pulling fluid back into the cardiovascular system. So that pressure is about 30 millimeters of mercury. So again, we have 55 millimeters of mercury forcing fluid out, and then we have 30 millimeters of mercury of pressure bringing fluid back in. So this opposes filtration because it's pulling fluid back into the glomerulus. And then the third pressure to take into consideration is the capsule fluid pressure. Now, with the capillary bed and the systemic capillaries and the pulmonary capillaries, they don't have a wall that surrounds those tissues. So the fluid can then just spread out once it leaves the systemic or the pulmonary capillaries. But when it comes to the renal glomerulus, there is a wall around it, and that's the renal corpuscle or Bowman's capsule. So the Bowman's capsule creates its own fluid from the pressure between the glomerulus and the capsule. So again, the capsular fluid pressure, it's hydrostatic pressure inside Bowman's capsule, and it opposes filtration that adds up to about 15 millimeters of mercury. So if you take the 30 millimeters of mercury of the colloid osmotic pressure and the 15 millimeters of mercury from the fluid that opposes filtration, add those up, you get about 45 millimeters of pressure, millimeters of mercury pressure. That is still a lot less than the capillary blood pressure. So you'll see that whenever your systemic blood pressure changes, it changes the blood pressure through the glomerulus. And if the kidneys will allow that to happen, then as your blood pressure rises and lowers, it's going to affect urine production. Now, there is a lot of useful information on this slide alone. It talks about the glomerular filtration rate, known as the GFR, things that affect the GFR. I'm probably only going to call it GFR from here on out because apparently I'm having trouble saying glomerular. So GFR, there are two parts to GFR. First, there's the net filtration pressure. That's whenever we take in consideration the three pressures we talked about in the previous slide. The capillary hydrostatic pressure, the capillary osmotic pressure, and then the Bowman's capsule hydrostatic pressure. That's the net filtration pressure. And then we have to look at the filtration coefficient. We talked a little bit about that way back in chapter 5. And this usually stays the same. It's the surface area of the glomeruli, the capillaries available for filtration, and then how permeable are the filtration slits. They should stay about the same. So we're not going to talk much about filtration coefficient. Now, the GFR stays relatively constant, and it's because we can change other factors that affect the flow of blood through the glomerulus. It has to do with the afferent and the efferent arteriole. If we remember back where the afferent and the efferent arteriole are located, the afferent arteriole is bringing blood into the glomerulus, and then the efferent arteriole is where blood leaves the glomerulus. And now let's talk about resistance. Remember, resistance opposes flow. So what increases resistance through a blood vessel usually has to do with the diameter of the lumen, the radius. Remember, the radius has the biggest effect on resistance, which then either opposes or increases flow. So we're going to look at how do we affect the blood flow through the glomerulus and how it affects the GFR. If we increase the resistance in the afferent arteriole, so increased resistance, we're talking about vasoconstriction. Vasoconstriction of the afferent arteriole, and it decreases the filtration rate. Well, how does that make sense? It's because we're limiting how much blood enters into the glomerulus. So less blood that enters the glomerulus, then less filtrate will be formed, so the filtration rate goes down. But what if we increase the resistance in the efferent arteriole? The efferent arteriole is going to be the arteriole that leads away from the glomerulus. So if we vasoconstrict the efferent arteriole, then we're going to increase the hydrostatic pressure inside of the glomerulus. So that would increase the filtration rate. Now, if we decrease resistance in the afferent arteriole, well, how do you decrease resistance? By vasodilation. So if we vasodilate, the afferent arterial, which means we're increasing the flow of blood into the glomerulus, then we're going to increase the filtration rate. And then if we decrease the resistance in the efferent arterial, what does that mean? That means the efferent arterial is now vasodilated, so more blood can leave the glomerulus. So if more blood can leave the glomerulus, then that will decrease or lower the hydrostatic pressure, so that decreases the filtration rate. Or if we look at this, talking about the glomerular filtration rate, those three pressures that affect the glomerular filtration rate, the net filtration. The hydrostatic pressure, forcing fluid out, right there, is about 55 millimeters of mercury. And then we have bringing pressure, or the osmotic pressure, bringing fluid back in. The colloid osmotic pressure is 30. So that's bringing fluid back into the glomerulus. And then we have hydrostatic pressure of Bowman's capsule. So that is the pressure of the fluid. That is the fluid that's going to be located, and I'm going to kind of color it in right here. This fluid here is bringing fluid back into the glomerulus, and that would be the 15 millimeters of mercury listed right there. Overall, though, we have a net amount of filtrate being formed, or net amount of pressure, and that's going to be, if we take the amount that's coming out from hydrostatic pressure and subtract how much is coming back in from osmotic pressure and subtract how much is going back in from the hydrostatic pressure of Bowman's capsule, then that will give us the net filtration pressure. Let's take a quick look at what happens if we change some of these pressures. How does that affect the filtration rate? What if there is an increase in the hydrostatic pressure of the glomerulus? How would that affect the filtration rate? If we increase this hydrostatic pressure, then we're going to increase the glomerular filtration rate. All right, what happens if we increase the colloid osmotic pressure? I'm just going to do COP for that. If we increase the colloid osmotic pressure, which is the pressure coming back into the glomerulus, then that's going to lower the filtration rate, right? Because it opposes the filtration rate. What if we increase the amount of pressure inside of Bowman's capsule? So if we increase the Bowman's capsule pressure, how's that going to affect the filtration rate? Then it opposes the filtration rate, so if that goes up, the other goes down. All right, hopefully I haven't totally confused you on that, but you have to get that down. What happens if you increase the hydrostatic pressure of the glomerulus? Well, then you increase the filtration rate. Well, how is the filtration rate affected if you increase the colloid osmotic pressure? Well, then that would decrease the filtration rate. Well, what would happen if you increased Bowman's capsule pressure? Then that would decrease the filtration rate. And then the same would be true for the opposite. What if we lower the hydrostatic pressure inside the glomerulus? Then that would lower the filtration rate. What if we lower the colloid osmotic pressure? Then that would increase the filtration rate. What if we lower Bowman's capsule hydrostatic pressure? Then that would increase the glomerular filtration rate. Typically, the glomerulus can auto-regulate its own filtration rate, which means it keeps it within a normal range, as long as the blood pressure stays within a normal range. So if we look at this graph, we have the mean arterial blood pressure on the bottom, the x-axis, and then we have the glomerular filtration rate, how many liters per day, on the y-axis. So 180 liters per day is the normal amount of filtration rate, well, per day, as long as the mean arterial pressure stays between around 82 to around 180. So between 80 and 180, as long as your mean arterial pressure, your MAP, stays within that, then the filtration rate stays the same. However, if the blood pressure gets too high and the mean arterial pressure gets above 180, then the filtration rate will go up. Or if your mean arterial pressure drops below 80, then your filtration rate will drop as well. This is a good picture demonstrating what happens when you increase the resistance of the afferent arterial, or if you increase the resistance of the efferent arterial. So if we look at this upper picture right here, blood going into the afferent arterial and into the glomerulus, creating a filtration, setting the GFR, and then blood leaving through the efferent arterial. You'll see that the diameter of the afferent arterial is naturally and normally larger than the efferent arterial, and that is because we're losing filtrate. through the glomerulus, so the efferent arteriole is not going to be as big as the afferent arteriole. All right, if we look at this picture down here, if we increase the resistance of the afferent arteriole, which means we've increased vasoconstriction of the afferent arteriole, then we're limiting the amount of blood that goes into the glomerulus, and then the GFR, the filtration rate, goes down because it decreases the hydrostatic pressure of the blood inside of the glomerulus. Then if we look at this other picture, if we increase the resistance of the efferent arteriole, which means we vasoconstrict the efferent arteriole, that means the blood that's entering through the afferent arteriole is building up the pressure inside of the glomerulus. So your hydrostatic pressure inside the glomerulus goes up, so that means your filtration rate goes up as well. So vasoconstriction of the afferent arteriole decreases the filtration rate, and then increasing the resistance on the efferent arteriole increases the filtration rate. Now what about if we let the pressure off? If we took pressure off of the afferent arteriole and vasodilated it, then more blood would be entering into the glomerulus and would increase the filtration rate. Or if we open up and vasodilate the efferent arteriole, it's going to increase the amount of blood leaving the glomerulus, and so that would decrease the filtration rate. And those are the things that we highlighted a couple of slides earlier. So how does the nephron auto-regulate its filtration rate? And there are three methods. There's the myogenic response, there's tubuloglomerular feedback, and then there are hormones that can affect the filtration rate. That's how it can regulate itself. So the myogenic reflex has to do with the smooth muscle of the afferent and the efferent arteriole. They automatically will constrict whenever there's been a stretch. So if we increase the amount of fluid going through the afferent arteriole, the stretch causes contraction of the vascular muscle. That's one of the things we looked at with blood vessels. Whenever blood vessels have a lot of blood in them, more than normal, then it expands the lumen and it stretches the smooth muscle, and stretching of the smooth muscle will open up the calcium channels. Calcium goes into the cell and causes a contraction. With the tubuloglomerular feedback, it's a paracrine control. Well, what does paracrine mean? That means there's going to be a secretion that affects the cells that are neighboring to the cell that is releasing the chemical messenger. And the apparatus used for tubuloglomerular feedback is called the juxtaglomerular apparatus. On the next slide, we're going to have a picture, so we'll have a little visual aid of what's going on with that. But basically, there are cells called the macula densa cells. Macula densa cells, they detect ions, sodium chloride in the filtrate, and then eventually they will trigger the release of renin, and renin has all kinds of effects. And we'll look at the picture here in just a second. Then we have hormones that affect the smooth muscle like epinephrine and norepinephrine. If they vasoconstrict the afferent arteriole, then blood will bypass around it, and that's how we can bypass the kidneys when you're in a fight or flight mode or the sympathetic nervous system mode. If you remember from anatomy, part of the ascending limb of the loop of Henle, right before it becomes the distal convoluted tubule, passes right between the afferent and the efferent arteriole. There are certain cells located right here inside of the ascending limb of the nephron loop where it becomes the distal convoluted tubule. These cells are called the macula densa cells, and they're real close to these cells that are between the afferent and the afferent arteriole, and those are the granular cells, the juxtaglomerular cells that are going to produce renin. But the macula densa, it detects and measures the amount of sodium chloride passing through this part of the nephron loop. If there's an increase in the amount of sodium chloride passing through, then that means that the filtration or the pressure inside of the fluid inside here is too high, or the amount of fluid passing by here is too high. So if that happens, then the macula densa senses an increase in sodium chloride, and then it will vasoconstrict. This is the paracrine secretion, the paracrine effect. It makes the afferent arteriole constrict, so it limits how much fluid is entering into the glomerulus, and it lowers your filtration rate. This is a pretty easy thing to follow. If we look at number 1 and go down, if your filtration rate increases, then the flow through the proximal tubule increases, which means the flow coming up from the loop of Henle will increase, and then the macula densa cells will detect that increase, and then it will create vasoconstriction of the afferent arteriole, so it limits how much blood is entering into the glomerular, so filtrate's going to be made, and it lowers your filtration rate. All right, we've looked at filtration. Now let's look at reabsorption. Remember that reabsorption is the movement of fluid or ions from the external environment of the tubule lumen back into the internal environment, either the peritubular capillaries or the vasorectal capillaries. Now, reabsorption can be active or passive. Remember the difference between those. Passive means it's going to go down its concentration gradient at some point. Active means it's going against the concentration gradient. There are two methods of reabsorption: transepithelial, transcellular, which means it goes through both surfaces of the apical cell. So it goes through the apical surface of the cell, crosses through the cell, and then out to the basolateral surface of the cell into the tissues. And then there's the paracellular pathway, and that is when it passes between the cells, and that had to do with passing through the cell junctions. Or if we look at this picture, and we have the tubule lumen on the left-hand side here, which means this is the external environment. So we're showing how these ions or fluid can go from the external environment into the extracellular fluid, which is internal environment. And then these cells are the cells of the tubule. So sodium, if it's going to go through the cell, it's going to pass through the apical surface of the cell. This is going down its concentration gradient at this point, so all it has to do is open the sodium channels. Well, now we want to get that sodium into the extracellular fluid. So in order to pass that sodium into the extracellular fluid, there has to be some sort of active mechanism right here. Could be using the sodium-potassium pump. And we have certain anions. Anions can pass directly through the cell, or they can follow the sodium, and they can go through to the between the cells in the paracellular pathway. Water, there are water channels called aquaporins, and they can open up into the cell and then pass through the basolateral surface, or water is small enough it can pass through the cellular junctions between the cells. This is showing you using the SGLT channel. We've looked at this in a couple of other different chapters. SGLT stands for sodium glucose transport. Remember, it's a co-transporter, and this is a secondary active mechanism. Glucose will bind to the receptor as well as sodium. Sodium binds first, which then changes the affinity for glucose. Then glucose can attach to the SGLT. Then they both go into the cell. Sodium will go down its concentration gradient. Glucose is going up the concentration gradient. And then when we get the basolateral surface, glucose will open up a facilitated transport mechanism. This is a passive mechanism, and then glucose will go down its concentration gradient into the interstitial fluid. And then we have the sodium-potassium pump, which is going to be an active mechanism. It's primary active mechanism of moving the sodium back out of the cell. And again, this is the sodium-potassium channel. Anytime we have protein-mediated transport, we have a transport maximum, and we're going to have saturation. With saturation, that means we are using the gates at their maximum rate of transport. If we only have two entry gates of glucose into the cell, and they're constantly being used, we can reach a maximum amount of saturation, which means we can only transport so many of those glucose molecules into the cell, so we can reach a transport maximum. And this will come into play when we look at glucose. We don't want any glucose in the urine, so we're going to reclaim as much glucose as possible. Now, glucose is going to leave with the filtrate because it's dissolved in the filtrate. That can't be avoided. But what the kidneys will do is reabsorb as much glucose as possible. But their gates can reach a maximum if the filtrate is just flooded with glucose. And this is what happens with diabetic patients. They have so much glucose in their system, it's in the filtrate, and the filtrate is trying to reclaim as much glucose as possible, but it can only do so much. So if it's completely flooded and they reach their transport maximum, that is when you get glucose in your urine. Let's look at the pressures and see what's happening here with reabsorption in the peritubular capillaries. Let's follow hydrostatic pressure first. The hydrostatic pressure inside of the glomerulus, that's the blood pressure of the blood inside the glomerulus, as we mentioned earlier, is around 55 millimeters of mercury. that favors filtration of fluid out into the tubule. But by the time it loses its filtrate, the amount of pressure that's going to go into the peritubular capillaries equals to about 10. Why is it so much lower than at the glomerulus? It's because it lost 20% of its filtrate. So now the hydrostatic pressure in the peritubular capillaries is around 10. Now let's look at the osmotic pressure. The osmotic pressure of pulling fluid back into the glomerulus is 30 millimeters of mercury. So now the direction of fluid is back into the capillaries. So whenever the osmotic pressure reaches the peritubular capillaries, it is the same. Well, why is it the same? It's because we didn't lose any proteins. The proteins stay in the circulation. So the osmotic pressure stays the same. So now if we look at the hydrostatic pressure again, it's at 10, and the osmotic pressure is at 30, then the driving force is pulling fluid into the peritubular capillaries around 20 millimeters of mercury. So the hydrostatic pressure is higher in the glomerulus, that's the driving force forcing fluid out, and the osmotic pressure is higher at the peritubular capillaries, which will reinforce reabsorption. So we've talked about filtration and we've talked about absorption. The last process that creates urine is secretion. So secretion is moving ions from the internal environment straight into the external environment. The two main ions involved in secretion are potassium and hydrogen. Now potassium control, that's important when it comes to your resting membrane potential. Remember back when we have hyperkalemia and hypokalemia. In hyperkalemia, we have high amounts of potassium in the blood. We can excrete excess potassium into the filtrate, put it into the external environment. And then the hydrogen ion has to do with your pH, and we'll look at that in the next chapter. Excretion is anything that's going to be excreted to the outside. So excretion is going to be anything that's going to happen after the filtrate leaves the collecting duct. So before, in other parts of the renal tubule, we had reabsorption and filtration, or reabsorption and secretion. Anything left over after the filtrate leaves the collecting duct is going to be excreted through excretion. So how do we find out how much is being excreted? First, you take the amount that's being filtered out, and then you subtract how much is reabsorbed, add back in the amount we're putting back in through secretion, and that will give you the excretion rate. Micturition is the process of eliminating the urine that is built up into the bladder. Once the filtrate leaves the collecting duct, it is urine, and you cannot change the composition of urine once it leaves the collecting duct. Where does it go after that? After the collecting duct, the urine will then go into through the renal papilla into the minor calyx, and then the minor calyx led to the major calyx, and then the major calyx led to the renal pelvis, and then the renal pelvis lead to the ureter. The ureter is the long tube that goes from the kidney all the way down on the back side of the abdomen into the pelvis, and then the ureter leads into the bladder. Now there are two sphincters or valves that control the flow of urine from the bladder. There's an internal sphincter, and then there's the external sphincter. The internal sphincter is involuntary, and it's at the base of the bladder between the bladder and the urethra. The external sphincter is located in the pelvic floor muscles. So internal sphincter is between the urethra and the bladder. The external sphincter or valve is that part of the urethra that goes through the pelvic floor muscles. The external sphincter is voluntary. So let's go to the next picture. Remember, micturition is under the parasympathetic nervous system control. Now, as the urine starts to fill into the bladder, which is what they're showing in this picture right here, when it gets to a certain point where it's full, it sends a signal along the stretch receptors, which then goes to the spinal cord, and it creates the micturition reflex. It'll send a signal back, and then it'll keep a tonicity to the bladder. And then it sends a signal to the internal sphincter. The internal sphincter will open. It's involuntary. You don't have to control it. Doesn't have to worry about it. But once the bladder is full enough, then the internal sphincter opens, and now the urethra starts to fill, and it stops at the external urethra, or the external sphincter of the urethra, which is voluntary. So now there are signals around here, and it'll keep the external sphincter closed until you can reach the appropriate place to open the external sphincter. Once you open the external sphincter, then urine can be relieved or be released into, well, wherever you happen to be. All right, this is the end of, or this is all I'm going to talk about in chapter 19 on the kidneys. Chapter 20, which I will release later this week, has to do with balancing water in the system, balancing the amount of hydrogen ion in the system, which will control your pH, plus some other ion balance as well. Email me if you have any questions.